DNA to RNA to protein. How are nucleic acids and proteins related? Making proteins depends on gene expression. When a gene is expressed, its DNA code is copied into an RNA transcript. RNA transcripts are then translated into proteins. The DNA to RNA to protein process is called the central dogma of biology, meaning that all of life's activities depend on this information flow. The rest of this presentation will review the basic biochemistry of proteins. The central dogma will be covered in the genetics presentation. Proteins. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and a little sulfur. Small proteins are also called peptides, and larger proteins are also called polypeptides. Proteins are chains of amino acids. Polypeptides are folded and gain function based on their unique three-dimensional shapes. Only 20 amino acids are used to construct all of Earth's proteins. Proteins are the machines, receptors, immune molecules, fibers, cytokines, transporters, and messengers of cells. Most genes encode proteins that carry out specific functions of living things. Components of amino acids. Amino acids include an amino group, a carboxylic acid group, and a side chain, an R group, all bonded to a central carbon. Each unique side chain, R group, determines the biochemical properties of its amino acid. So all amino acids have the amino group, which is the NH2, the carboxylic acid group, which is the COOH, and the side chain, the R group, is just abbreviated as R here because it is unique amongst the different amino acids. Amino acids. In this diagram, the variation in the 20 R groups is highlighted in blue. The arrangement of these R groups in a 3D folded protein gives a protein its unique biochemical functions. Protein shape and structure. Shape is critical to the function of a protein. A protein's shape depends on 4 levels of structure. Primary, that's just the sequence of amino acids. Secondary, the common folding patterns within the backbone. Tertiary, the final 3D shape of the polypeptide. And quaternary, the joining of more than one polypeptide. Protein function depends on shape. The code for each protein lies in a gene. A change in a gene may lead to a change in a protein's primary sequence, the sequence of amino acids. A change in primary structure may alter secondary, tertiary, and quaternary structure. A change in structure is a change in shape, which will have an impact on its function. Sickle cell disease results from a single amino acid change in the hemoglobin protein. Changing the primary structure impacts the quaternary shape of hemoglobin, the shape of the red blood cell, and its ability to carry oxygen. We see a normal blood cell on the left, that donut-shaped cell that could just glide through blood capillaries very easily, versus the sickled red blood cell which cannot flow as easily. Denaturation. The shape of a protein often depends on its environment. Changes in temperature, pH, and salt concentration, for example, can cause changes in folding. When a protein unfolds due to extreme changes in the environment, it may not be able to refold. Permanent unfolding of a protein is called denaturation. Denatured proteins have lost function permanently. Frying an egg causes denaturation of the proteins in the egg white. They will not regain function. Functions of proteins. Proteins have thousands of different roles in an organism. 8 common categories of function are shown. Structural proteins have function in hair and the cell cytoskeleton. Contractile proteins are part of muscle and other motile cells. Storage proteins are sources of amino acids or ions, and defense proteins can serve as antibodies or blood cell receptors. Functions of proteins. Proteins have thousands of different roles in an organism. 8 common categories of function are shown. Transport proteins can be hemoglobin or membrane channels. Signaling proteins include hormones and membrane receptors. Enzymatic proteins can regulate speeds of reactions. Machine proteins are motor proteins that move organelles. Microtubules move chromosomes.